Class 3 amino acids. GABA, glycine, glutamate, aspartate. Right? Class 4 gases like nitric oxide. Right? NO, nitric oxide. Or carbon monoxide, CO. Sometimes also H2S. Okay? So these, I'm not sure about this, so this, okay, so these two, okay? So, nitric oxide, carbon monoxide are gases. Right? So, these are small molecule neurotransmitters. They are involved in active responses of the nervous system, like in detecting sensory signals or motor signals. So, when we feel something by touch, okay, the signals go through the synapses by way of these neurotransmitters, small molecule neurotransmitters. Or, when we move our body, the motor function is caused by the release of these type of neurotransmitters, small molecule neurotransmitters. But besides these neurotransmitters, we have also neuropeptides. They are different actually. So you know that each protein is made in endoplasmic reticulum, right? So you know that we have ribosomes. Ribosomes convert mRNA, translate mRNA into proteins. So all of this happening in the cytoplasm of the cell body, so on. But neurotransmitter is released from the axon terminal, right? So we have the nucleus of the cell in the cell body. Nucleus generates mRNA. mRNAs come out of the cell of the nucleus, right? It binds to the ribosome. Ribosomes translate it into proteins, right? Protein is packed in some vesicles through the endoplasmic reticulum and Golgi apparatus, right? Then the vesicle goes through the axon to axon terminal. So they are stored in the axon terminal. So these are neuropeptides, okay? So they are smaller than proteins. They are actually composed of a few amino acids, right? So they are not huge like proteins. They are smaller, so that's why we call them neuropeptides. So you can see a long list of these neuropeptides. I come back to you, okay? So you can see some of these names. You may be familiar with some of these. For example, you should know prolactin. Yes, or luteinizing hormone, LH. Pyrotropin, right? Like TSH, right? Growth hormone, vasopressin, oxytocin. So these are hormones. You have this in endocrine physiology. Remember that?
Yes, sir.
So, what's the difference of the hormone and neurotransmitter? Hormone is secreted by some glands into the blood and go through the blood to everywhere in the body, right? But neuropeptide is not like this. It is released from the presynaptic cell onto postsynaptic cell, right? So there is no circulation here. There is no blood here. So it's directly act on the postsynaptic cell. But hormone is released, for example, from the thyroid gland. It goes through the blood all over the body. Okay? But the chemical is the same. So, for example, the same oxytocin, which is released by the gland into the blood, can be present also in the synapse. Okay? So the structure is the same, but the origin is different. Right? Here, the oxytocin is made by a presynaptic cell, not by the gland. Okay? So try to not memorize, but be familiar with this. Okay? It's important for me that you know the differences between these neuropeptides and the small molecule neurotransmitter. For example, I may ask you that which one is a neuropeptide? Okay, A, dopamine, B, serotonin, C, nitric oxide, D, oxytocin. So this one is a neuropeptide. Okay? But usually I give you some more complex options. Okay? So try to remember this. So insulin also here. Okay? Insulin can be released from a gland. You know the gland, right? Beta cells of the pancreas, right? So they are released from the pancreas into the blood. It causes, movement of the sugar into the muscles, into the cells, right? So some neurons also in the brain release insulin. Okay? Some neurons in the brain generate insulin, they pack them in some vesicles, and they release insulin onto other cells as a neurotransmitter. Right? Did you get the difference and the point? Okay, so.
Sir.
Yeah. First, you. Okay, you.
Sir, I was saying you referred about the slow and rapid transmitting. Does it depend on, and does it refer to the synaptic delay, how much it takes, or
for neuropeptides, you mean?
Yeah.
No, it's not. It's not the same. No, it's different. I'm gonna talk about this now. Okay. You. Okay. So no other questions?
If we put the neurotransmitter in the synaptic clip, there is no need to stimulate the neuron.
There is no what?
Potential will generate.
Potential generated.
If we artificially put the
artificially.
Yes,
So if you artificially put the neurotransmitter
yes,
into the synaptic route, it can bind to the receptor and can have some function. So we do this in our everyday life.
action potential will generate.
Yeah, action potential is generated. For example, you may take a lot of drugs, right? For example, you may consider a patient which has a Parkinson's disease, right? And they use a drug called L-dopa. This is a drug, okay? Medicine, okay? So, this one is converted to dopamine inside the brain and go to the synapses and activate the postsynaptic receptors. So instead of having dopamine from the presynaptic cell, we are giving the patient the precursor of the dopamine. Okay? Or, for example, you can consider a patient with major depression. Okay? So there is lots of norepinephrine and serotonin inside the brain of these patients. Right? So you can give them some drugs that activate the synapses. So imagine that you have a norepinephrine synapse here. Okay? So this one releases norepinephrine onto this one. So now there is a lack of this neurotransmitter in the brain of the patient with major depression. Okay? So instead of giving norepinephrine to this patient, you give some drug that actually block return of the neurotransmitter to the presynaptic terminal. So actually, after the release of the neurotransmitter, most of them come back again into the axon terminal. So in this way, they can end the function of the neurotransmitter, okay? So we call this reuptake. So we have reuptake of the neurotransmitter back to the axon terminal, okay? So we may have some drugs like TCA, tricyclic antidepressants. So this drug blocks this one, the reuptake process. So norepinephrine is released, it cannot come back. So it remains in the synapse. So this way, the action of the norepinephrine in the synapse is activated and it's prolonged. Okay? So instead of increasing the concentration, you are increasing the action. Okay? So we can artificially manipulate the function of the synapse in order to cure a disease or treat a patient. Okay? So, let me go on.
Yes.
Okay? So, I told you that there is a synthesis process for neuropeptides, right? mRNA, translation into proteins, hacking of them into the vesicles, then they go through the axon to the axon terminal. We call this axonal streaming, right? Then they reach the axon terminal, and they can be released after the arrival of action potential, right? Usually smaller quantities of these neuropeptides are released. So this is a keyword here. Which neurotransmitter is released more? Small ones, right? They are generally 1,000 or more times as potent as a small molecule neurotransmitter. So which one is more potent? Neuropeptides, right? They can cause much more prolonged action. Okay? Each of them has actually prolonged their action, neuropeptides. So neuropeptides have prolonged action. So this is a table for comparing these small molecules and neuropeptides. Okay, so classic transmitters, peptide transmitters. These, small molecules are made in the nerve terminal, but neuropeptides are made in cell body. Small molecule neurotransmitters are made in their active form, right? When they are made, they are active. If you release them, they can do function, right? But neuropeptides should be cleaved, should be segmented. They should be broken down in order to become active. This happens in the vesicle, okay? Small molecule neurotransmitters are stored in small, clear vesicles, but neuropeptides are stored in larger vesicles that are electron dense under the electron microscope, right? They are released, the classic neurotransmitters are released in synaptic cleft, but neuropeptides can be released some distance from the target. For example, I have a lot of markers here, right? Some of them are working. So, for small molecule neurotransmitters, we have active zone, right? They can be released from active zone for a small molecule neurotransmitter. But neuropeptides actually sometimes are released in another mode, like this. Imagine that we have a synapse like this. Okay, it's not end here. The axon continues. Then we have another one here, right? And we may have another one here, we call them varicosities. Okay? So neuropeptides can be released from here. They go and can be released from here. So they may have several targets. And they are released some distance from the cells. So they're not so localized like this one. Okay? So they may be released some distance from the postsynaptic what? Postsynaptic, I cannot find it, okay? Postsynaptic cell, okay? So, another difference is that the action of classic transmitter is ended by reuptake, I told you. but the action of the neuropeptide is ended by proteolysis. So it's a peptide, but it's released. There are some enzymes that degrade these peptides, okay? Or they can be diffused to some far distances for the lasers, okay? So proteolysis and diffusion are mechanisms that can end the action of the neuropeptide. And the last one is that classical transmitters have short action and short latency of action and short duration of action. But neuropeptides have some longer delays, long latency of action, and the action persists for some time. Okay? So any questions so far?
Can you explain that one? The proteolysis, what's the difference?
Proteolysis? Yeah. So you know that proteins can be degraded by some enzymes.
Okay.
I'm asking what the difference. Can you explain the difference?
Difference between these two, you mean?
Yeah.
So, when a neurotransmitter is released, it binds to the receptor and it has some action. This action should be ended after a while. It cannot be continued forever, okay? So, the ending or the completion of the action is different for classical neurotransmitters. They usually come back to the axon terminal. For example, I told you for norepinephrine. They are released, norepinephrine, right? Then we have some proteins responsible for reuptake of these chemicals. So this way, norepinephrine is removed from the synapse, right? But for neuropeptide, it's different. For example, imagine that insulin is released here. There are some proteins, right, that degrade the insulin here. So the enzymes convert the insulin into the amino acids. Okay?
Sir, what is the exact location of proteins in synaptic cleft or
Yes.
synaptic membrane?
No, in synaptic cleft. Also on the postsynaptic cell. You know, between the cells we have a matrix, right? So it's a network of proteins like collagen, everything. Okay? So in this matrix we have some proteolysis. Okay? Proteases. So they are not inside the cell actually. They are out of the cell. Okay? So how many minutes do we have? 14. You look tired? No, we continue. This figure shows a concept called co-release of the neurotransmitters. So I told you that we have two separate groups of neurotransmitters, classical or let's say small molecules and neuropeptides. But sometimes they are released together from a cell, right? In the first example, you have the co-release. So you can look at these vesicles. In each of these vesicles, we have two different neurotransmitters, the green one and the purple one, right? So two small molecule neurotransmitters are connected together in a vesicle. So when action potential arrives, we have the exocytosis of the neurotransmitter. So both of them are released at the same time, right? So we call this co-release. So a cell can release two different types of neurotransmitters. Actually, when I was a student, in our book, it was mentioned that every neuron has only one specific type of neurotransmitters. It's not possible for a neuron to have two different types of neurotransmitters. But now in your book, it's different, you know? So it's a new discovery, actually. Some neurons can release two different neurotransmitters at the same time. So we call this co-release, right? So sometimes in other cells, we have co-transmission. It's different from the co-release. In co-release, two different types of neurotransmitters are in a single vesicle, okay, at the same time. But here, we have two different populations of the vesicles. Neurotransmitter the green neurotransmitter is inside one vesicle, the purple one is inside another vesicle, right? But they are not released together when the action potential arises, because their sensitivity for the calcium and for the action potential is different, right? Imagine that the green one is more sensitive to calcium, and the purple one is less sensitive, right? So when only one action potential arrives, some calcium goes into the cell, they activate the proteins of the cytosis, okay, the green one is released. But the calcium here is not enough for the release of the purple one, because it needs more calcium, right? So in order to be released, there should be several action potentials. So when you have several action potentials, several course of calcium entrance happens, right? So they increase the concentration of the calcium more than the previous one, okay? So we have more calcium. So this vesicle can also be released, okay? So we call this co-transmission, but with differential calcium sensitivity. Got it? Okay? Another type of co-transmission. An axon is divided into two synapses. In one of them we have the green one, in another one we have purple, right? They are released together, but at different locations, right?
Yes.
So they have the same sensitivity to calcium. So both of them are released, but not in the same synapse, in different synapses, right? So try to differentiate between these two. These three actually, co-release, co-transmission with differential calcium sensitivity, and co-transmission with spatial segregation, right?
So, this vesicle could have same, for example, a small neuropeptide, a small neurotransmitter or peptide at the same time in a vesicle.
Yeah, they can be, both of these can be small neurotransmitters, but we have also neurons that release, for example, small molecule neurotransmitters and neuropeptides. Usually neuropeptides are less sensitive to calcium. Okay, so we need more action potential for the release of the neuropeptides.
Sir, the function of two neurotransmitter releasing the co-release is same or different? The function of the neurotransmitter?
Yeah, no, they are different. Yeah, so one synapse can do two different functions at the same time. One of them by one neurotransmitter, another one by another. Okay? So sometimes, actually we have different types of activity in these synapses. Sometimes one of the neurotransmitters amplify the action of the other. Okay? We have this, for example, when we have release of the neuropeptide together with a small molecule neurotransmitter. Like, to give you an example. So imagine that from this neuron, you have small molecules and larger molecules, right? So this one is glutamate. This one is called orex, a neuropeptide. They are released from the neurons of the lateral hypothalamus. Okay? This glutamate activates some neurons. For example, in the ventral tegmental area, another area in the brain. Okay, but the action of the glutamate is amplified when orexin is also released. So orexin makes the VTA neurons more sensitive to the glutamate. So sometimes it's like this, but some other time, together with glutamate, we have, for example, dopamine, maybe. Okay, so dopamine have some functions, glutamate have some other functions. They are different, but they are released together. Okay, so we have this in the CNS. Okay, and this is very new actually. So a lot of works are doing on this concept nowadays. Okay.
Sir, these neurotransmitters are released at the same time.
Yeah.
Is it possible to have two action potential in a neuron at the same time?
Yes. Why not? One action potential arrives, and immediately after that, another one arrives. Okay, so one neuron may generate 10 action potential in a second, or it may generate, for example, 100 action potential in a second. Okay, it's possible actually.
So it won't be simultaneous. It can be like one after another, but it won't be simultaneous.
Yeah. So, for example, in this one, you mean this one,
Yeah, exactly.
right? So, if only one action potential arrives, only this one is released, right? But if, for example, 5 action potential come to the action terminal each after the other one, then we have the release of both this one, because one is enough for that, and we need 5 action potential for this one, so we have simultaneous release of these two. Okay? So, I can continue to talk about the synapse if you like that.
Yes.
Okay, but I think that some of you maybe feel tired. I don't know. It's up to you.